Hello. Today I'm going to be talking to you about virus, specifically the term virus and viroid and virion, as they have been developed this past summer of 2020 for the purposes of use in the virus infectious disease ontology and the COVID-19 infectious disease ontology. So first, I'm going to talk about some of the starting points for the development of the term and definitions associated with it, some potential problems that have arisen with other terms in the OBO Foundry that are nearby. I'm going to talk a bit about the Baltimore classification, which I find to be an elegant way to organize virus terms. And finally, we'll end with definition of virus and some related terms. So first, we'll start here with the NCBI taxon, which is a very common exhaustive list of terms that one finds in the biomedical field or the biological domain generally. Indeed, VIDO, that is the virus infectious disease ontology, imports terms from the NCBI taxon where possible, like for instance virus and virion. But look, the taxon does not provide much ontological structure. It doesn't really provide textual logical definitions, but if we're going to align with OBO Foundry principles, we're going to need this stuff. We're going to need a robust hierarchy or some sort of like robust ontological structure, textual definitions, and logical definitions. If we focus on the first as terminological, the robust hierarchical structure with respect to the terminological content, we can see how this sort of gap leads to some problems. Look, this is because many researchers working in ontology import terms automatically using the software Ontophox, which is incredibly useful. It's developed by the Hague Group, maintained by them as well. Basically, you can just go over to ontology or an ontology that exists, and you're going to import terms without much reflection. It's an automated process. So what's happening in this case is people are going over to the NCBI taxon, which has a very minimal taxonomy structure. It's just kind of like they want like a species term, for instance, for a virus, and so they import blindly from the taxon. But when they do that, it imports like every single thing that ranges from kingdom to species, and this makes the resulting ontology quite difficult to navigate. Here's an example. So if somebody wants to import a term, like look down at the bottom, like Homo sapien, can import this term from the taxon, but it also imports all of these other terms, all the way up to eukaryota. Look, we probably don't need all of these terms if we're going to be talking about Homo sapiens. This was pulled from a Brucellus bacteria or a Brucellus infectious disease ontology. It seems unlikely that in like using or talking about Brucellus in human patients is going to need to make a lot of reference to, say, bilateria or chordata or anything like that. Of course, this could be found and should be found in other ontologies, but this sort of violates the modularity principle. Like we don't need all of this information in the Brucellus ontology. It makes it quite difficult to navigate. Like clicking through all of this is not fun. Now, boo is my ultimate evaluation of this. On the other hand, and in addition, textual definition and logical definitions, they're lacking from the taxon. Since we want to follow best practices for ontology development, we're going to need to come up with textual and logical definitions. And we also need to add more robust structure, not obviously the sort of structure we saw when importing from Ontophox, but basically figure out a different way to maybe a more elegant, I think more elegant way to arrange virus terms within logical constraints. Now, this leads to the Baltimore classification. So virologists sometimes or frequently will talk about classifying viruses in an exhaustive way in one of these 7 groups based on the sort of genetic makeup. Here's a visual representation of the various groups. You see over here, we've got like herpes simplex is in group 1. We've got some common like familiar viruses like influenza in group 7, and poliovirus over here in group 4. This incidentally is also where you find a coronavirus. It is a group 4 virus. And basically, let me get back. What this means, if we're just focusing on the group 4, this positive sense single-stranded RNA, classification. This is a characterization of, say, polioviruses or coronaviruses based on their genetic makeup. And what it means is that during the transmission process or the translation or replication process, starting with sort of positive RNA here, the virus, in order to complete its replication cycle, it has to go translate this material into negative RNA, which is then translated into positive messenger RNA. And so there are multiple steps that are required by any virus falling in that grouping um, before it can replicate. Now, on the parvovirus, for instance, over here, group 2, it starts with positive sense DNA genetic code, and so it basically can just kind of hop over to this double-stranded and then pop over to the positive sense mRNA. You can see influenza at the bottom just has to make one step, just jump straight from the negative sense to the positive messenger RNA, and similarly for the reoviruses and rotaviruses, they have this double-stranded in group 3, and they can pop over to the messenger or the positive sense messenger RNA. So again, this is an exhaustive classification of viruses. Any virus, you can find it in one of these groups. It's defining the terms in with respect to what you might think of ontologically speaking as the nature, the material nature of the entity, and that namely its genetic makeup. And it's a rather elegant and clean way to both capture what it is that makes viruses up in an exhaustive, clear way, but also by explaining in part what they can do, or at least providing a foundation for what viruses can do specifically. So with this in mind, we start here and we define virus as an acellular structure. This term was defined in a previous or in another talk, which was basically like an arrangement of units. I would advert you or point you to back to that talk on acellular structures for more information about why. But look, a virus is an acellular structure with an RNA or DNA genetic material. This uses host metabolic resources for RNA or DNA replication. Alongside these Baltimore classification, or alongside this term, the other terms from the NCBI taxon that correspond to Baltimore classification terms can be imported as well. Like, for instance, the positive sense single-stranded RNA virus. These are viruses with a genetic material that's encoded in a single-stranded RNA that can be translated directly into proteins. Now, here is a sort of visual representation in the protege editor of some of the definitions and some of the relationships. So you see virus here, and under that you have double-stranded DNA retrovirus. You got positive, we've highlighted here positive sense single-stranded, and subclass of this is going to be coronavirus. In fact, we use it as an example of use. And this corresponds, as I mentioned a minute ago, to this membership in this group 4, which, by the way, again, Baltimore classification group 4 is an exact synonym we're using for positive sense single-stranded RNA virus. Now, note, if anything, this is not an incredibly long, exhaustive taxonomy all the way from kingdom to, coronavirus or picornavirus. Like, we're not like importing all the terms you would need in some very extensive hierarchy to get here. We just, 7 groups, all viruses. This is a clean way to classify each. Also, we have this term virion. I think it's worth discussing. We define it as a virus in its assembled state, consisting of genomic material surrounded by coding molecules. Now, this is worth talking about because the term virion is not very clearly used in literature. I can point you to the paper and we should discuss this in more detail, but some use it in a way that suggests virion is to virus as like a human gamete is to a human. That is not exactly right. It's not a sperm cell. That's not what a virion is. It's supposed to be a completely assembled virus particle. Virion, some say, is a virus is a human student who's to a human. This would make it more like a role, like a virion is only a virion whenever it's like in a certain environment, much like the way a student is only a student when it's in a university or enrolled in a university. That doesn't seem right either, because what makes a virion is it's sort of like material structure, not where it is. Some say a virion is to a virus as a human child is to a human. This is, in fact, the way we're using it. This seems the more consistent, more compatible way of using the term across literature, even if people are speaking loosely. A virion does seem to be like, a stage in the development of a virus or a stage in a virus. Viruses, they go through several stages. They have like latency stages, but they also have fully assembled stages. And like virus particles or components can be identified or with those stages while nevertheless continuing to be a virus. And so it's warranted in classifying them as a subclass of virus for that reason. We can also say that some viruses are not like well developed or fully completely assembled because some viruses don't and this is useful because some viruses don't replicate successfully. They might lead to virion mutants, and these are frequently associated with their own diseases. So you could have like a mutated coronavirus that is not associated with COVID-19, for instance. It could systematically replicate poorly, and because of that, infect or nevertheless maintain its ability to infect hosts and be associated with some other set of symptoms or some other disorder or some other disease that is caused specifically by that version of the virus. We provide terms for this, specifically a cellular structure having some arrangement of viral components like a capsid or a viral DNA or RNA that is clinically abnormal. So in this case, the virus itself is experiencing some abnormality. It is disordered. Okay, so just to conclude here, Vito imports terms from existing ontologies where possible, but adds textual and logical definitions in alignment with OBO Foundry principles so we can provide a clear and concise foundation from which ontologies covering specific infectious viral diseases may extend.